Hey everyone, and welcome. If you're a first-year med student, I know biochemistry can feel like a mountain to climb. So today, we're going to tackle a really crucial topic, the metabolism of phospholipids. And trust me, these aren't just fats. They are literally the architects of every single cell in your body.
Just let that sink in for a moment. Every single one of the trillions of cells that make you is defined and protected by a membrane built from these amazing molecules. But, they do so much more than just build walls. Let's dive into how.
So here's our game plan. First, we'll properly meet these architects and figure out their key jobs. Then, we're going to go inside the cell and check out the assembly line where they're built and recycled. After that, we'll see what happens when that assembly line breaks down, and this is where we get into the real clinical stuff. And finally, we'll zoom out and see how this all fits into the body's huge metabolic map.
All right, first up, let's meet these cellular architects. What in the world are phospholipids, and why is understanding them going to be so important in your future medical practice?
Okay, the one word you absolutely have to know here is amphipathic. It sounds complicated, but it just means these molecules have a split personality. They've got a hydrophilic head that loves water, so it happily faces the watery environment inside and outside the cell. But then they have these hydrophobic tails that hate water, so they tuck themselves away from it, creating this incredible self-organizing barrier. That simple property, that's the foundation of life as we know it.
Now, not all phospholipids are created equal. They basically come in two main families, and it all depends on their backbone. You've got the glycerophospholipids, which are built on a pretty simple glycerol frame. And on the other hand, you have sphingophospholipid, which use a more complex backbone called sphingosine. It's kind of like the difference between building a house with a wooden frame versus a steel frame. Both create a structure, but they have different properties and are used for different jobs.
And their job description is huge. I mean, look at this. Obviously, they form the basic structure of membranes, but they're also the main ingredient in lung surfactant, which literally keeps our lungs from collapsing. They're heavy hitters in cell signaling pathways. They're a key part of the lipoproteins that taxi fats through your blood. They even provide the raw material, arachidonic acid, to make molecules that control inflammation. These things are the ultimate multitaskers.
So let's meet a few of the star players. Phosphatidylcholine, or lecithin, is a real workhorse. It's everywhere in membranes, and it's the key player in that lung surfactant. Then there's phosphatidylethanolamine, which you'll find tons of in your brain and nerves. Phosphatidylinositol, or PI, is absolutely vital for sending signals across the cell membrane. Cardiolipin is a special one. It hangs out almost exclusively in the inner membrane of our mitochondria, the cell's power plants. And finally, Sphingomyelin, which is essential for insulating our nerves in what's called the myelin sheath.
Okay, section 2, the assembly line. Let's pull back the curtain and go inside the cellular factory to see how these critical molecules are actually built, broken down, and managed.
Now, the synthesis pathway can look a little intimidating in a textbook, but honestly, it really just boils down to 3 main steps. First, you got to activate one of the starting pieces by tagging it with a high-energy molecule. Second, you form a reactive middleman, an intermediate like CDP-choline. And third, you just transfer that activated piece to its partner, and boom, you have your finished phospholipid. So remember, activation, intermediate formation, and transfer. That's the whole logic right there.
And where's all this action happening? Well, these assembly lines are humming along at full speed in the smooth ER of some really important organs. The liver, your body's metabolic command center, is a huge site, but so are the brain, the lungs, and the lining of your intestines, all places where strong, functional membranes are non-negotiable.
Of course, anything that gets built eventually needs to be taken apart, and for that job, we have the phospholipases. These enzymes are like the cell's expert recycling crew. They carefully disassemble phospholipids, which is super important for remodeling membranes, creating signaling molecules, and just general cellular housekeeping.
And this recycling crew doesn't just smash things with a sledgehammer. They have a very precise set of tools. Each phospholipase, A1, A2, C, and D, acts like a pair of molecular scissors, making a specific cut at a different spot on the molecule. Now, pay very close attention to phospholipase A2. This one is a big deal. When it snips the fatty acid at that second carbon, it very often releases arachidonic acid. This is the starting gun for the entire inflammation cascade, and it's the exact pathway that powerful anti-inflammatory drugs are designed to shut down.
All right, now we get to the core of why all this biochemistry matters to you as a future doctor. We've seen the architects, we've toured the assembly line, but what happens when that perfectly organized system goes wrong? This is where the textbook hits the bedside.
So, what are the consequences when this incredibly delicate assembly line breaks down?
Let's take a look at a major clinical example: respiratory distress syndrome.
This is a truly devastating condition we see in premature babies. Because their lungs just aren't fully developed yet, they don't have enough of that lung surfactant we talked about. And without it, their tiny air sacs, the alveoli, just collapse every time they breathe out. It becomes a desperate, exhausting struggle just to get oxygen. And what's the direct biochemical reason? It's a failure of the assembly line to produce enough of one specific phospholipid we've already met, dipalmitolecithin. This molecule is the main ingredient of surfactant. When it's not there, the consequences are immediate and life-threatening. This is a perfect straight line from a single metabolic pathway to a clinical emergency.
Now let's look at the other side of the coin. What happens when the recycling crew fails to do its job? Niemann-Pick disease is a tragic genetic disorder where cells can't break down a particular phospholipid. This causes a toxic buildup of sphingomyelin inside the cell's recycling centers, called lysosomes, leading to severe damage in the liver, spleen, and brain.
The problem comes down to one single faulty enzyme, sphingomyelinase. It's one of the tools from our phospholipase toolkit. When that enzyme is broken or missing, the recycling of sphingomyelin grinds to a halt. It's just a powerful, tragic reminder of how one broken part in that degradation pathway can cause a devastating, system-wide disease. Okay, for our final section, let's zoom all the way out. Let's place everything we've just learned into the bigger picture and see how these pathways connect to the entire metabolic map of the human body. Like any good factory, this whole process is very tightly regulated. Its speed really depends on three key things. First, supply and demand. You need the raw materials, precursors like fatty acids and choline. Second, you have hormonal control. Signals like insulin can tell the factory to ramp up production. And third, you need power. The whole thing runs on energy from molecules like CTP to fuel those critical activation steps.
So, if we step back and organize all their jobs, we can really put them into three main buckets. Structurally, they're the fabric of our cells. Functionally, they do things like acting as lung surfactant or sending signals. And they also have a protective role, constantly working to repair our membranes and defend against damage. And this is a really important point. None of this happens in a vacuum. Phospholipid synthesis is tightly connected to other pathways. In fact, it shares a key intermediate, diacylglycerol, or DAG, with the pathway that makes triglycerides, our main stored fat. This creates a critical fork in the road, a metabolic crossroads where the cell has to decide, do I use this DAG to build new membranes, or do I use it to store energy for later? So this all leaves us with a huge, forward-looking question. We've seen how tiny errors in these pathways can cause terrible diseases. So how can we be smart about it? How can we design future drugs to specifically target the synthesis or the breakdown of certain phospholipids for therapeutic good? The potential to treat everything from inflammation to cancer to neurodegeneration is just immense. Thanks for joining this explainer.